Welcome to Molecular Biology Techniques Session 1. Fine. Let us go to the first slide. Now, in order to do any molecular biology techniques, the first thing that you need to do is you need to fractionize the cell, which means you need to take a tissue and separate it into its component cells, and from the cells you need to take out the DNA. So these are some of the steps which are used for cell fractionization. So you can see here there is homogenization. First the tissues or the cells are taken in a container from where they are homogenized. Whatever is homogenized is called as a homogenate, after which you subject it to centrifugation. So here differential centrifugation has been given as one of the methods, because centrifugation is one of the ways in which you can separate the individual homogenate into its individual components. Now, these are some of the equipments that are used for the purpose of cell fractionization. So here you can have the advantages of simultaneous disruption and homogenization. Some of the equipments you can see from one particular company: tissue ruptor, tissue lyser, tissue lyser 2. So for example, you can see it is used for human or animal tissues or for plant tissues. It can be used for bacteria and yeast, and these are the kinds of instruments that are used for cell fractionization. So once you do the cell fractionization, now you have an active compound from which you can extract the DNA. So what are the difficult things and what are the easy things to take out from the cells? So easy ones are the cells, yeast, some liver tissues, spleen tissues, kidney tissues, etc. Difficult to lyse are usually fatty tissues, brain, skin, adipose tissues, and fibrous tissues, muscle, heart, trachea, and lung. So these are just some examples to make you understand that different tissues have different challenges when you are trying to homogenize or fractionize the cells. Understood? Fine. Now, once you have the cell fractionization or you have the homogenate and you have centrifuged it, and now you have some component from which you can extract the DNA. So how do you isolate and purify the genomic DNA? Genomic DNA means whole DNA. So you can see in the picture that you first need to break down the cell wall and membranes. You centrifuge and separate the solids from the dissolved DNA, after which you do a precipitation step, and finally you will get a dissolved DNA. So here on the right side of the screen you can see a flow chart of the steps that are involved in DNA extraction. So any source tissue extract, I showed you the different instruments that are used for homogenization. So any tissue extract you take and you lyse the cells. Once you lyse the cells, then you remove the membrane lipids. After that, you denature the proteins and remove them, and then you remove any other contamination. Mainly what you are doing here is you are removing all the contamination from the cells. So once you remove all the contamination from the cells, you will end up with what is called as purified DNA. So in some steps you need to extract RNA. So in that the protocol will be different. But here it is extraction of DNA. So finally you will end up with DNA. The DNA you have got from what? You have got from the purified tissues. You have not got it from the purified tissues. The tissues have actually been homogenized and disrupted, and they have been taken as cells. And from the cells you have again homogenized them, and you have converted the homogenate that you have centrifuged, and from that centrifuged processed part only you are doing this entire process. So it is not easy to say directly DNA is extracted from the tissues. There are a lot of steps that are involved in extraction of the DNA from the cells. So here you can see another another representation of the DNA or RNA extraction given by a company. So on the top right side you can see the usually a spin column is used for doing this work. So this is the modern way of doing it, how the DNA is extracted. And you can see a graphical representation of the amount of DNA or RNA which is being extracted from the tissues or the cells. Okay. Now, once you have the DNA, there are a number of molecular biology techniques that you can subject it to. One of the most important techniques is polymerase chain reaction, which you are going to use for amplification of the DNA for different purposes. Okay. So, here you can see that you have a template DNA and you have a target. Okay, this target is this here within the template DNA. Here is the target, and this is what you need to make multiple copies of. So usually this DNA is a double stranded molecule. So when you are subjecting it to heating, what will happen? The DNA double stranded molecule will denature. it will separate into its individual single strand molecule, and when you lower the temperature, they will again anneal. But during the annealing stage, you introduce a primer strand and you introduce single nucleotides which will latch on to the single strand at the particular place, and then it will amplify. So this entire process, if you keep on repeating again and again, each part is called as a cycle. If you keep on repeating again and again, you will observe that you will get more and more copies of the double-stranded DNA molecule. So you start off with one single double-stranded DNA molecule and you end up with a lot of copies. So in this diagram, you can see that at each cycle, the number of copies are increasing in exponentially. First you have 4 copies, then in the next cycle you have 8 copies, in the next cycle you have 16 copies, and finally you have 32 copies. So at the end of 40 cycles, you will have 2 to the power of 40, which is 1000 billion copies. So this is the range of amplification that you can get, and this is why polymerase chain reaction is being carried out. So this is a very simple representation of how the polymerase chain reaction is there. First you have a double-stranded DNA molecule. In the first cycle, the DNA will denature at 94 degrees Celsius into 2 single strands. Primer will anneal to the single-stranded DNA. You can see one primer annealing here, one primer annealing here. So one is moving in the 5' to 3' direction, one is moving in the 3' to 5' direction. So annealing will take place at around 55 degrees Celsius. Then you have the Taq polymerase enzyme which is needed for polymerization. That polymerization process will extend the range of the primers from the 5' to 3' and 3' to 5'. So you have a forward primer and you have a reverse primer. And finally, the target DNA is doubled. And this entire process is called as one cycle. This cycle is again repeated. So during the process of every cycle, you have the repetition and the amplification of the DNA molecule. Now, this is the photographic representation of a thermal cycler, and this is the kind of tube in which the thermal cycler reaction is done. Whatever is mixed here, that is the DNTP, that means dinucleotide, and the DNA template, the DNA polymerase enzyme, which is usually Taq polymerase enzyme because it can withstand the high temperature. Then the primers. All of this is called as the PCR master mix, and they are kept in what is called as PCR reaction tubes, and it is kept inside a thermal cycler. A thermal cycler is nothing but an equipment which will carry out this cyclic process of heating and cooling the reaction process. So here you can see during the first cycle, the 95 degrees denaturation is done for 5 minutes, and for the next 30 to 35 cycles, 95 denaturation is done for 1 minute, 55 degrees primer annealing is carried out for 1 minute, 72 degrees Celsius extension for 1 minute, or for 1000 nucleotides. That is the assumption given. So one cycle, when it is completed, 72 degrees final extension for 5 minutes. So this is like different types of programming that you can give in the thermal cycler for carrying out the polymerase chain reaction. So apart from the regular polymerase chain reaction, these are some of the common types of PCR. There are different methods of PCR, but these are the most common types of PCR. So first you have the regular PCR, which is the polymerase chain reaction. Then you have what is called as RT-PCR. RT-PCR means reverse transcription polymerase chain reaction, where you use the reverse transcriptase enzyme. Then you have the qPCR. qPCR also called as RT-PCR, but you should not get confused between real-time PCR and reverse transcriptase PCR. So usually real-time PCR is referred to as qPCR. So qPCR means real-time polymerase chain reaction. Then you also have as the qRT-PCR, or the RT-PCR or the qPCR is combined as a singular technique. So let us look at RT-PCR versus the conventional PCR. The major difference is here, instead of Taq polymerase, you are using reverse transcription, and you start off not with a DNA molecule, but you are going to start the reaction with an RNA molecule. So the RNA is extracted from the cells instead of the DNA extraction. So once you extract the RNA, you get what is called as cDNA. cDNA means complementary DNA. So once you have the complementary DNA, how do you get the complementary DNA? You get the complementary DNA by using what is called as reverse transcriptase enzyme. When you use the reverse transcriptase enzyme, the RNA molecule is converted into cDNA. Now you have cDNA, which is nothing but a double-stranded DNA molecule. Now you can subject it to the regular amplification procedure. So you can see that you can carry out the PCR after this step, and you will get an amplified cDNA component, which is a representation of the original RNA strand that you started with. So this kind of PCR is called as, what it is called, RT-PCR or reverse transcriptase PCR. So what can it do? What is so special about qPCR, which is real-time PCR, which is something different from RT-PCR. RT-PCR is reverse transcriptase PCR. qPCR is real-time PCR. So generally in normal PCR reactions, what is the problem is, we will do the PCR. After that, we have to run the gel and ascertain the amplified amplicons. But in qPCR, the real-time PCR, what you can do is you can observe graphically whether the reaction is proceeding or whether the amplification is taking place and to what extent the amplification is taking place. This has a number of applications. So as you can see in the slide here, the qPCR can monitor the progress of the amplification when the process is going on. So a real-time PCR can perform detection, analysis and quantification of the sample. Quantification means you can measure how much of the gene is present in the particular sample. So detection, what it means? It means finding out the presence of targeted gene sequence, which is assured by the presence of the amplification curve, which I will be explaining shortly. Then quantification. Quantification of the targeted DNA in a sample can be done by using the cycles needed to obtain the threshold value of detector and PCR efficiency. Next, these are some of the components of the real-time PCR detection system. Now, this is a very simple example. Usually it is done, the detection is done because of the help of fluorescent dye. A fluorescent dye is added along with the PCR mix so that the reaction is taking place. The fluorescent dye will latch on to the different parts of the double stranded molecule, and the amount of fluorescence detected will directly correspond to the amount of amplification which is being carried out. This is how real-time PCR is able to detect the amount of amplification that is happening. So fluorescent dyes are of two types. They are non-specific or they are strand-specific. So non-specific or DNA binding agents are known as cyber green. There is a very famous fluorescent dye called as cyber green 1. And strand-specific or hydrolyzation-based assays use TACMAN or molecular beacons. So these are the different methods used for as fluorescent dyes for the detection of the amplicons for real-time PCR. So here you can see the application of the different types, cyber green and Taq polymerase. So there are two types of markers used for this purpose. TACMAN probe, and TACMAN probe has what is called as a reporter dye and a quencher dye. You can see how it is working in this particular slide. Molecular beacons, you can see how it is working here. Now, this is a very important graph. Please pay close attention to this graph. This is actually the real-time PCR reaction when it is done. You will get a graph here where the fluorescence is appearing as a graphical line. On the left side, you have the fluorescence intensity or the accumulation of the amplified product. On the bottom slide or on the x-axis, you can see the number of cycles of the PCR reaction. So you have 10 cycles, 20 cycles, 30 cycles, 40 cycles. So during the cycles, you can see that gradually the fluorescence will increase and it will reach a plateau phase. So there is an initial exponential phase and there is a non-exponential phase. So the amount where there is a minimum threshold line beyond which wherever it is crossing, it is called as the CT value. So the CT value is very important. The CT value means that cycle number of PCR reaction where the fluorescence will cross the threshold line and the CT value is low, then initial copy number of template was high, so it crossed the threshold line earlier cycle. So for example, if it crosses it at the 10th cycle itself, it means the number of genes that we are searching for is probably very high in the sample. If it is crossing very late towards the 30th cycle only, it is going amplified, then it means the number of genes in our initial sample is very low. So if CT value is high, then initial copy number of template is low, so it crossed the threshold line in late cycle. These are some other examples of the line. You can see the fluorescence increasing as it goes, and if you take the temperature curve, it is also important to take the temperature curve. and this temperature curve is further studied to get the melt curvatures. So these processes are used to more accurately determine how much gene is being quantified and what gene is being quantified. Now, let us take an example of a case study for the direct application of gene expression analysis by real-time PCR. So, real-time PCR is often used to find out whether a gene has been expressed in a particular sample. So, here on the right side, you can see the graph, the exponential phase, the non-exponential phase, and where the material will cross the CT value. So, let us take two samples, sample 1 and sample 2, and both of them how much they are containing one particular gene called X, gene X. So the CT value for sample A when it was run with qPCR was found to be 20. The CT value for sample B was found to be 22. What was the conclusion is? Conclusion is that the gene X is expressing more in sample 1 than sample 2. But how did we come to this conclusion? Is it possible to calculate accurately whether it is possible? Let us see how it is done. So here you have another example where you have 1000 cancerous cells and 1000 normal cells and want to check the gene expression difference in these two types of cells of the gene P53 by real-time PCR. So in both of them you have the gene P53, okay, in normal cells and you have in cancerous cells. So how many cells you are taking? You are taking 1000 cancer cells and 1000 normal cells. Now when you run the PCR, you will get a CT number based on the graph. The CT number of the P53 ones were for normal cells it is 15. For cancerous cells it is 12. But how do you take it just like that? For that you need what is called as a reference gene. The reference gene used in this case is GABDH. So this is the reference gene. So at the same time of running the sample, the reference gene is also taken. So at the time of reference gene, the CT number for the reference gene is 16.5 in the case of normal cell and 15.9 in the case of cancerous cells. So I am hoping you are able to understand. The CT number is higher here, CT number is lower here. So how do you calculate the triple delta CT value? Step 1. Calculation of the delta CT, difference in CT values. So CT delta CT of normal is equal to CT of the target gene. What is the target gene? The target gene is P53. We want to find out in which cell it is more expressed. So CT value of the target gene minus CT value of the reference gene, which means here it is 15 minus 16.5, which is minus 1.5. Then delta CT of the cancerous cell is CT target of the target gene minus CT of the reference gene, which is 12 minus 15.9, which is minus 3.9. This is the first step to calculate the delta CT values between the normal and the cancerous cell. Step 2 is delta delta CT, which is equal to delta CT of the cancerous minus delta CT of the normal cell. So here you get the minus 2.4. Next, step 3 is 2 to the power minus delta delta CT value, which is nothing but 2 to the power of minus minus 2.4 equal to 5.3. So there is a 5.3 fold difference of gene expression. This is known as fold difference of gene expression. So how do you conclude this solution? You have to tell that in cancer cells, the P53 gene is expressing itself 5.3 times more than the normal cell. So that is the meaning. Directly you can see here itself when you are looking at the CT values, the cancerous cell is at 12, CT value is 12 and the normal cell CT value is 15. Here itself you can conclude that it has reached the CT, it has crossed the threshold value much earlier than the normal cell. But you need to do all these calculations and prove with the help of a reference gene. Only then you can say confidently that the cancerous cell is expressing that particular gene more. And what is the difference? How many times is it increased? For that you need this triple delta T calculation. So let us go through the formula once again. Delta CT is equal to CT of the target gene minus CT of the reference gene. Delta delta CT is equal to delta CT of the sample minus delta CT of the control. So finally this is the formula to find out the fold difference of the gene expression. So let us go through the entire protocol once again. The cells, you take the RNA, then you convert it into cDNA, then you subject it to RT-PCR, When you do RT-PCR, you will get fluorescence either by cyber green or by TACMAN or by molecular beacons. But you will get what is called as a CT value. That is the value at which it crosses the threshold line. At how many cycles it crosses the threshold line. Thank you very much.